Our next speaker is going to be Manuel Kaulich from Vivilion GmbH, and he's going to tell us about the next frontiers in functional genomics, design, execution, and analysis of combinatorial CRISPR screens. Take it away, Manuel.
Thank you. Yeah, let me just start by saying it's great to still see so many awake faces. And I'm representing Vivilion here today. But what I decided to do, instead of giving a sales pitch, because this is the sponsor talk, I want to take that time and make a step back. And what I mean by that is we have, in the first 3 talks of the sessions, we've all know our CRISPR screening experts, so we know how they work, and we know what great results can come out of this. But what I want to talk about a little bit is quality control. So how do we actually know that the experiments we're doing are of sufficient quality, of sufficient depth, that we can actually analyze them and that we have certain confidence in the hits? And the example that I want to use is, we're all aware of like monogenetic screens, so 1 guide per cell or 2 guides targeting the same gene per cell, all monogenetics. But the example that I want to make is that the vast majority of phenotypes that probably we're interested in or that are disease-causing, they're not monogenetic. In fact, they're combinatorial. They are polytrait diseases. And so our attempt on solving those is by actually doing combinatorial genetic screens. And I want to take that as a theme for the quality control parameters that I'm going to allude to you. So what I want to do is, within the next 60 minutes is to tell you a little bit 4 snippets. The first one is I just want to bring everyone up to speed about what is sort of like the search space we are talking about. And it's gigantic. The second part is about how do these reagents need to be designed or can be designed so that actually meaningful biology is coming out. The third part, which I think is highly underappreciated, because we take computational biologists just for granted, is what are the analytical concepts that are behind a good hit calling. And the fourth part is I want to give you a snippet and how we use this actually in my own lab to actually identify combinatorial phenotypes. So I want to just start off by bringing everybody up to speed and say cellular genetics is complex. Okay, we know that. But the point I want to make is no gene acts alone. None, in fact. 10 to 30% of all single genes cause, sort of like in the knockout environment, cause a phenotype. And that's DepMap. We heard that before. You're knocking out a gene, an essential gene in a given cell line. 30% of the cases, it's essential. But what's more interesting is that 1 to probably 5% of paired knockouts are actually unexpected phenotypes. So what does that mean? That means by both of the phenotypes of these 2 genes individually, you cannot anticipate or you cannot expect or predict the combinatorial phenotype. And that's by definition a genetic interaction. So gene A non-essential, gene B non-essential, AB essential. That cannot be predicted just by single gene phenotypes. And then 1 to 5% of the cases, this holds true. And the numbers actually go up if we select for genes that are evolutionary conserved or related, like for example paralog genes, ERK, MEK, MAP kinase signaling, full of paralog genes. You knock out 2 paralogs that are essential for a certain pathway, you will get a phenotype. You knock them individually out, you don't see something. And I think on Friday we'll hear more about this by Trevor, who is going to talk about that probably with the paralog library. So just so that we are all now up to speed, I just want to say a few words about the search space so that we know how gigantic this actually is. And I want to start off by just saying, okay, what is the monogenetic search space? And we heard that. There's 20,000 genes in the human genome, but not all of them are expressed, obviously. In a given context, we're talking about roughly 10 to 11,000 genes that are expressed. And of course, we can use focused or genome-wide perturbations to identify those phenotypes in a given context, like DepMap has been done. You know, we can do knockouts, we can do base editing, prime editing, whatever, but it's all monogenetic in that particular search space. We can do even isogenic screens, compare a parental line with a knockout line, and then identify synthetic vulnerabilities. We can all of do that. But again, that's just one background. But the search space becomes even bigger if we consider context. So now we're talking about 1,000 cell lines of DepMap all in a sudden. So now that's gigantic. But it becomes even more complex if we consider that genes don't act alone. So we are talking about a combinatorial search space of 20,000 times 20,000 genes. Again, not all of them are expressed, right? So the possible search space in this particular case is 200 million combinations. Those excludes ABBA pairs, so we equalize those. And that can tell you something about genetic interactions, dual paralog essentiality, as I said earlier, but also protein complex predictions. If you're interested in this, we can talk off stage a little bit. But even here, that's just for a given context. You consider context, the search space becomes unimaginable. It's too large to handle, right? So we need to come up with smart designs, experimental smart designs, to actually have a laser focus approach to make means of that search space. How can we entangle that? And obviously the unfeasible approach is genome by genome. But there are other approaches, which I will come to in a second. And what that can tell you is, for example, higher order genetic interactions. Yeah, you get dual phenotypes in a context-specific manner. Or multi-paralog essentiality, for example. You do a screen of 2 paralogs in a context where the third one is not expressed, for example. Or protein complex predictions and now complex interactions within cells can be done. And multi-trait correlations, obviously. So genome by genome, I said, is too large to handle. But one can do query by genome. So one picks out maybe 10, maybe 20, maybe 50, maybe even 100 genes, generates a combinatorial library against the genome. That is a feasible experiment. But still, very large. So how can we further narrow this down? The other approach is obviously a fixed pair approach. You go in and you say A with B, C with D, E with F, and so on and so on. You have a focused approach, and paralog libraries are a great example of that. You have a predetermined combinations of guides. Obviously, you limit yourself because you're doing a biased experiment, but at least the search space becomes manageable. So I think this is sort of like the search space we're talking about. And obviously there are challenges associated with this. And one of which, the biggest one, is the sheer dimension and scalability, but I think we can manage this by sort of like what type of libraries, how do we design them? The analytical strategies, so what concepts are there? This is something I want to share with you. And then how you can prioritize gene combinations for actually taking or identifying meaningful biology. So let's start with the first one. So combinatorial libraries. I already mentioned this, so I can run over this really quick. So there are 2 general scenarios. One library design is called symmetric, in which basically the dx and the y in your dual matrix are identical. Yeah? So that's a symmetric all by all. The other possibility is you have an asymmetric form, in which you have a few genes by all, or you have a few by a few. Yeah? So this is fixed pair in this case. Now, talking a little bit about controls when you generate those reagents, I think we learned a lot from these 1,000 genome-wide screens that have been done, monogenetics. So we always include core essential genes, non-expressed genes, enhanced proliferator genes, and that's just for QC to assess whether your screen worked. That's not useful for hit calling. That's only for assessing the quality of your screen. Of course, we also include NHTs, like non-human targets. They don't have a target, so you want to see a null phenotype, basically. Reporter genes, they don't even exist in your background, for example. Again, just null. Ideally, you have positive controls, depending on the screen you're doing. And all of these controls that should be in a library, they hold also true for combinatorial libraries. But there are a few more things that we have to consider, because we need to think about the analysis in order to include the correct controls in such a reagent. And there are 3.5-ish essential controls. The first one is, in a combinatorial library, you should have single versus dual targeting. What I mean by that is, when you target an essential gene with 1 guide, in the same library, you should also have the gene targeted with 2 guides. And I will come to that later, why this is important. The second thing is, you want to control for the gRNA position. You have multiple guides, there are different positions, even in an array, they're in a different Cas12 or Cas13, there are different positions, right? So you want to control for that. And that's because you're using different scales, different tracers, using different promoters, and so on and so on. So you want to make sure that there's no positional effect, which is a huge contributor to noise in such an experiment. Self-targeting, so that means that relates to the first point that I made. So include core essential genes and enhanced proliferator genes that are targeted with 1 guide and with 2 guides. In a second, I will explain this. And of course, if possible, positive controls, like, for example, context-independent paralogs, for example. Oh, I have 2 more minutes. Wow, that was really okay. Wow. Then I have to speed up a little bit. Okay, I can jump over this probably. So we do a lot of QC. Sorry, I really want to show the use case because I think it's really cool. So I'm just going to jump over this. Jens Ole told me 20 minutes. Really? Okay. Ah, well, that is not my fault. Okay, anyways. Okay, I run over this. Sorry for this, guys, but I really want to share the use case with you. You're interested in the analytical concepts, please talk to me. I think it's crucial. And I hate that, obviously, that we don't have enough time to skip over this. So, to move over this. So anyways, so here's the use case. So what I want to show with you is a combinatorial library in which we took 450 cancer druggable genes. So those are the drug targets of everything that is approved, and then phase 1, 2, and 3. It's a multiplex library. Each gene is targeted with 2 guides. So roughly, the library has a diversity of 1 million guide combinations. We use this library in a cellular model that expresses a Cas9 and a Cas12. The Cas12 is constitutive, and the Cas9 is DOX-inducible. So what that means is one of the guides will be active right from day 0, and the second guide will be active only once we add DOX. Now, we can use our cell line, and we can hit DOX right away, and then we have a combinatorial screen, right from the beginning, dual editing. But we can also wait 2 weeks, and then add DOX. Now we have a directional screen. A comes first, then comes B. And since it's a symmetrical library, we not only have A to B, we also have B to A. So we can ask really cool questions, like, for example, iterative drug treatments. Do we see phenotypes in which the order of genetic events from A to B is different from B to A? So we do lots of QC on these data sets, and that's what I wanted to allude to you before, but obviously for the interest of time, I couldn't do that. And then what we do is first we look at the genes where we do not expect a directional phenotype, which are paralogs. And in this case, what we show here is the delta between the log fold in this case and an anchor edit, so means directional screen versus the dual edit screen. And you see that for the most part, they're very similar in their effect. So there is no directional effect. Yeah? But we can focus on genes, and this is now looking at the ones that have a very big delta, that have a very strong effect. And one of which, I don't have a pointer here, but probably too small to see on the very right bottom, it's a heat shock, which is a paralog together with a DAP. And it's particularly interesting because the DAP protein, USP7, is an essential gene. And in this particular screen, we identified a paralog dependency, a directionality in which this essential gene becomes non-essential. And if you're interested in what the molecular mechanism of this is and how we use this directional phenotype to basically explore more of that, then please come and let's talk a little off stage. So yeah, sorry that I ran over time. I thought a little bit more, but okay. So I have summary I probably can skip. I have to thank the Vivlion team, obviously. So if you're interested in these reagents or screening service or data packages, please talk to us. But I also have to thank my lab in Frankfurt because they have done the work that I showed you with the use case. And with that, I thank you for your attention.
So for the sake of time, we will take only one pressing question, if we have. Anyway, I'm promising you, I'm going to come and ask a question during the drink time after that. So thank